Hello, everyone, and welcome to today's live broadcast, Achieving Higher Throughput with Multiplexed Phage Display, presented by Dr. Thomas Hengstl, Software Product Manager, Molecular Devices. I am Alexis Krauss of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by ForteBio. For more information on our sponsor, please visit fortebio.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Thomas Hengstl. Dr. Thomas Hengstl is currently a product manager for software and compliance with molecular devices. Prior to joining the company, he received his PhD in biophysics and biochemistry through a joint international industrial program between the University of Natural Sciences Salzburg and Genentech Incorporated. The development of the multiplexed phage display workflow described in the webinar was a product of his PhD program. Dr. Hengstl, you may now begin your presentation.
Okay, well, thank you, Alexis, for this nice introduction, and welcome to today's webinar about Multiplexed Phage Display, which is a rapid peptide discovery and screening platform. So, a little bit about myself, in addition to what Alexis told you. I did my bachelor thesis in Krems, which is in the lower Austrian part, as well as with a cooperation at Genentech, where I did the actual research lab. And the bachelor thesis was about developing peptide binders against sonic hedgehog. This is where I really started to get excited about phage display. In my master thesis, I validated an alternative antibody scaffold for the use on phage, which was on a different university, but again at Genentech. And for my PhD thesis, I actually put together my own project, which was Multiplexed Phage Display, which really optimized every aspect of phage display and used the latest and greatest technology to improve throughput, but also the amount and the detail of the data that was generated. Since 2016, after I finished my PhD, I'm now a product manager for SoftMix Pro, which is a compliance software provided by Molecular Devices. And the connection between me and ForteBio, which I have to point out here, is that ForteBio actually moved into the Molecular Devices conglomerate as a business unit. But my PhD work itself did not have to do anything with Molecular Devices or ForteBio. This was completely independent. And below you find my contact information. All right, let's get started. So, a little bit about myself, you already heard that, as well as ForteBio Molecular Devices. Now we'll give you an overview about the technology, so you know what to expect from this webinar. A brief introduction to phage display for those who aren't that familiar with this topic. And then we're going to have two main topics, which is the introduction of the technology itself by an example where I develop a peptide binding to LRP6. This work has already been done. I have deliberately chosen this topic or this protein because it has already been done and we knew at least partly what to expect. As well as the second topic, which focuses on the throughput. Since we're stating here to have a high throughput phage display technology, I will also show you an example where we have taken advantage of this technology to its full extent. And at the very beginning, we have obviously the Q&A session. All right, before we get started with the actual topic, we're pushing out a polling question to you. And what I would like to know from you is, what do you see as the biggest bottleneck in phage display workflow as you do it? We have 4 options: either library generation, phage panning, lead candidate characterization, or data analysis. All right. So, technology overview. What we're going to do is in Multiplexed Phage Display. So first, you start off with the regular phage display process. But instead of doing phage titration assays to basically evaluate the phage concentration, we're going to come in with the qPCR. This is nothing new. This has been reported in the literature. But what we are doing is actually, so we are using barcoded primers, as well as we've optimized the qPCR, so you can use the product from the qPCR straight for next-gen sequencing. Since we use barcoded primers, everything from one plate can now be pooled into one tube. It can be sequenced, and after the sequencing, you get the peptide counts, how many times one peptide was in the library. So don't worry if you don't understand everything at this point here, and hopefully you don't. But this is what we're going to go through in the webinar in a very close detail. And what we have found, and this is new, at least I haven't found it in the literature at the point that I was doing my PhD, is to actually calculate signal-to-noise ratio, which is usually done by spot ELISAs, from also sequencing the background in this well. Now we have the next-gen sequencing results, and typically what we see from there is a bunch of lead candidates. But we all know that the abundance of a phage clone in a certain library doesn't necessarily tell you about the binding affinity. So what we're going to do is, and this is also another technology I've developed during my PhD, is the strep-David infusion technology. And within a single day, and by ordering one single DNA primer, you can actually biophysically test your lead peptide on the octet system. And this is also extremely high throughput compatible. So ideally, by the end, you have next-gen sequencing results as well as biophysical data, which gives you a very clear idea of what's going on in your library and what your lead candidates are. All right, so let's get started for those who haven't really worked with phage display and didn't have a clue about what was going on in the previous slide. So phage display is, as the name says, a phage is a bacterial virus, and we use it as a display system. So we would like to display something on the surface of the phage. So as every virus, it basically you have a capsid as well as a DNA that is enclosed to that capsid and encodes everything that is required to assemble the virus. So what people found is you can basically clone the gene that's responsible for the capsid and add it on top of the original copy in there. So what's happening is you're getting a mosaic. So this means you can theoretically use this additional copy to get something into the phage and to have it displayed on the surface. So these are the two different systems. This is the 100% penetrant system, and this is called a phage-mit system. And we will be working with a phage-mit system here. So what do I mean you could take advantage of that? So what you can do is you can purify this phage-mit out of the phage, and by having a DNA library where every primer, and here we talk about a lot of different primers, like 7 to the power of 11 and higher. So these primers essentially encode peptides. So what you can do is you can basically align those primers to the phage-mit by a technology that's called concol mutagenesis. This is what you see here. So you have a single-stranded template, you anneal the primer, then you transform everything into bacteria, get the second strand, which has not been modified, digested, and you end up with a phage-mit where the new DNA and the changed DNA is incorporated. If you transform all of that into E. coli will produce the bacterial virus, where now the surface of the virus basically displays your peptide. And what's shown here in 5 copies is typically a library of 10 to the power of 10 modified phage. So you have quite a diversity in there. And ideally, depending on the length of the peptide, you encode the full amino acid spectrum there. Our target protein, as I said before, is LRP6. This has just randomly been chosen. I wanted to use something that has been phaged before in order to show the comparability between multiplex phage display and standard phage display. And to LRP6 itself, so LRP6 is a cell surface co-receptor of the beta-catenin pathway and has been shown to interact with DKK1. But we are not focused on the biology, but we are more interested in the technology here. All right, so let's go through the technology in detail and see how we actually detect peptides and develop peptides here. So our target protein gets coated onto the well. There are typically 3 different options how you can immobilize a protein for phage display. So either you directly coat it on the well, or you add a biotin, coat streptavidin, and use this as a linker to kind of prevent denaturation, which is happening in the well. Or you phage it in solution and pull down the phage complex and the protein complex together. Down here, you will see an illustration of the plate, what has been used for the phage display. And as you see, we only use one well at this point. And the green one's always the target reaction containing the target protein. And the orange one is the control reaction, which in our case is just a well that has been blocked with BSA. All right, so let's look at the molecular, what's happening on the molecular level here. So we have the protein coated on the well. And we have a control well that's coated with BSA. So the individual BSA molecules are not shown. Now you add your phage library. And the idea is, after an incubation period, so every phage that displays a preferable motif actually binds to your target protein. But there is also non-specific phage that hangs around on the well. So after you wash it away, what you would like to see is in the control well, non-specific phage that is bound to the well and BSA and pretty much anything that's also in the target well, with the exception of the violet phage here that is target specific. So theoretically, if you subtract those 2 from each other, you should only end up with the violet phage. So how do I know when I can stop the phaging process? Well, what you can do is a qPCR. So you take the elutions from the phage display and you conduct the qPCR. We're going to have a look at the results in a few seconds here. But if you don't see enrichment in the qPCR, what you would do is you would reinfect bacteria, you would amplify the phage, and now you get an enriched library. So everything that's not bound got washed away, and you only have the phage that got enriched, and they obviously multiply by the thousands. All right, but now here, how do we determine if actually there is more phage in our target reaction than in our control reaction? We do qPCR, and the qPCR is done as follows. So you have a reverse primer that binds downstream of your region of interest, which in our case is the peptide, but can be up to 400 base pairs. And you have a barcoded forward primer that binds upstream. And the little barcode, that's really just some DNA sequence that's unique. And we have 96 primers. So for each well, for each library, we have a separate primer. This helps us at the end to basically demultiplex the whole reaction again. So we have sequencing adapters, we have the phage map binding sequence, and we have the same thing on the reverse primer. So at the end of the day, what we would like to end up after the sequencing reaction is the sequencing adapter, a barcode that is specific for the well, so for the library, the 5' binding, the target region, the 3' binding, as well as the sequencing adapter. So in the qPCR itself, multiple things happen. So first of all, once we amplify them, what we would like to see is a delta CT, so a difference in where the binding curves are coming up. Because this actually means, so if the target reaction is coming up sooner, this means there must have been more phage in our target reaction than in the control reaction. And the only way this is going to happen is if there was target-specific phage. And another important thing up here is the plateau phase. So what we have done is we have tested out how low we can go with the primer concentration, and we keep the primer concentration the same for all wells. So this means, although the target reaction is coming up sooner, it hits the same plateau. So at the end of the day, all reactions in here have the same DNA concentration, which makes it really easy for us to pull them, because if there would have been a concentration difference, even at the end, we would need to adjust the libraries in order to get the same numbers of sequences from the next-gen sequencing. So this is the data, what we saw, and we've done that for every round. And although we already saw enrichment in the first round, so we probably could have theoretically stopped here, we continued and we reduced the amount of bait of target protein. And at some point, we just reduced it too much, and we didn't see that much enrichment anymore. However, so we took the round 4, but for this case, you could have already stopped for round 3, and we also analyzed these reactions. So the control definitely came up later than the target, which means there was enrichment, and we're good to go with the sequencing. So as I said, since we have this barcode, this unique barcode in here, we can actually pull this library from A1 together with the control library and any other library we have. So this means you really can fill up the full plate. And that it really works, we will see in the second application I'm presenting to you. So you take aliquots of essentially all of your libraries and you pull it into tube, and then you mix it. And then we are already good to go with the sequencing. Just a little overview, what's happening. So one piece of DNA gets encapsulated with a sequencing bead, and this is the ion torrent technology. Illumina works exactly the same, you just have to use different adapters. So then you do an emulsion PCR, the whole bead gets covered, and then you flush it over the chip surface, and in one pH-sensitive bucket, one bead covered with DNA ends up, and it's single-stranded. Next, you flush the nucleotide triphosphates over the sequencing chip, and obviously a primer is attached here as well. And whenever there is a binding event, from a biochemical perspective, the binding event of a trinucleotide results in the release of a pH, and this can be detected by the pH-sensitive layer since it's happening everywhere. What we get from the next-gen sequencing is a vast amount of sequences. So typically, we have somewhere between 5 and 50 million sequences, depending on the technology you need. What you usually need for your library is somewhere between 10 and 50,000 sequences per library, and that multiplied by on how many libraries you're sequencing. So the bioinformatics are fairly simple. We use the BioPython script for that, which I wrote myself. So you take the FASTQ file, which now contains all the sequences from your plate, and since we know the sequence of the barcode, we can now demultiplex the individual sequences into the files. Then we trim off the 5' and the 3' region, so we end up with the inserts, which are the peptides, and then we translate it. And then we take each peptide sequence and we count how many times we find it in the target library and how many times we find it in the control library. And from that, we can calculate a signal-to-noise ratio. So this means we don't have to do any phage ELISA anymore, but this is already our signal-to-noise ratio, and this works really nice. So you end up with a list of lead candidates of total abundance as well as the signal-to-noise ratio. And since we know that the abundance isn't enough, we have to biophysically characterize them. But here we don't have any DNA, so we need to synthesize our DNA. These were the results from the next-gen sequencing. We used 50,000, we analyzed 50,000 sequences per library. What you see is this peptide here has the highest, by far the highest abundance. This peptide here shows a high signal-to-noise ratio, suggesting that this is a non-specific binder, and the same for this peptide. The 14 highest ranked binders we used and characterized KD values. So how do we now get actually a peptide? How we produce a peptide cheap and fast? So what you do is you create another primer. But in this case, we set it up for in vitro translation rather than next-gen sequencing. So we add the 5' untranslated region, a start codon, the peptide, which in our case are now the 14 highest ranked binders. So we look for the peptide sequence and order this primer, as well as the streptavidin binding sequence. And the same for the reverse primer, but this one is universal. So you only need to order a forward primer for each peptide you would like to test. Then you do another PCR reaction. So and you use a regular POC 57, for instance, with a streptavidin insert. I would recommend reducing the GC content because even the GC content of streptavidin is extremely high, and you can get problems in the PCR. So you need to order a GC content reduced version. So now you do the PCR. So you end up with a double-stranded DNA product of the 5' untranslated region, a start codon, the peptide of your interest, and remember this is DNA, the streptavidin stop, as well as the 3' untranslated region. And this piece of DNA we're now going to use in an in vitro translation reaction. So once again, this whole process is still 96 well compatible. So it's very easy to test 96 peptides at a time for a very cheap price and extremely fast, because all you need is to order this primer here. So for one peptide, you need to order one primer. In the in vitro translation reaction, the double-stranded DNA template, and you can use straight PCR template. That's, of course, you need to purify it, but you don't need to do anything else. So you add it to a 5 microliter in vitro translation reaction, and this reagent can be ordered through NAB. And after 1 hour, the DNA has been transcribed into mRNA, as well as already translated into protein, and with an attached peptide. So you now have a tetramer of streptavidin, as it naturally is a tetramer, attached to your peptides of interest. And you have that for 14 different peptides. And also this is fully high throughput compatible here. All you need is a multi-channel pipette. And in the next step, we can use the ForteBio octet system to characterize all of those peptides in just 2 hours. So full 96 well plate can be run through in 2 hours. So you get biophysical data of 96 peptides in only 2 hours. And ForteBio offers different versions depending on your throughput, starting off with single biosensor measurement devices all the way to high throughput 384. The technology, how it is going to work is you have an optical layer, and you have immobilized molecules, which in our case are the streptavidin, the peptides fused to streptavidin. You have light coming in, and it's reflected, and it's in phase here. But if you increase the thickness of the layer, meaning a binding event is happening, there's a phase shift, and you can calculate the KD from that. So what's happening now is, with the biosensor, which is biotinolated, you only need to dip into the in vitro translation reaction for 1 second. And in 1 second, this extremely high affinity of streptavidin for biotin picks up enough material for you to go into the next well and measure binding to your target protein. And then you go into the dissociation well, and you can measure dissociation rates. And this is, once again, fully high throughput compatible. So it's no problem to do that with 96 peptides at a time. And after this experiment, you get your KD value. These are the KD values for the 14 peptides. We measured the blank tip and only streptavidin as a control, which are down here. If we now look at the summary, we can actually find some really interesting findings which have been, in general, have been reported in the literature as well. So for CNA KGC, this was the top 1 peptide in the NGS. And it still shows up as the top 1 peptide after the biophysical measurement. Also, number 2 was a very good binder in the micromolar range. Whereas number 3 is quite a surprise. Although it ranked number 17, it showed up as the 3rd tightest binder. And what's also very interesting is that this peptide down here, as well as this one, which showed a very high signal-to-noise ratio, didn't show binding to LRP6 at all. So this kind of confirms what we see from the signal-to-noise ratio. As well as this peptide, which was the 3rd most abundant peptide in NGS. This one didn't show binding either. And in order to confirm the biophysical data we got from the octet system, we also measured synthetic peptides. Although you see some difference, this is perfectly fine because we used the system to confirm the binding. So in order to summarize the findings for LRP6 here, is the highest ranked binder contained NAIK, which is a motif found in the murine DKK1, which interacts with the E1 unit of LRP6. So this makes sense. That 2 cysteines maybe stabilize the peptide and lock it down in a certain configuration. And the glycine probably was the one residue that didn't disturb the motif the least, because we used the Seramer peptide library. So the conclusions for MPD here, more is better. 70 out of the 100 most enriched peptides were not detected by picking 96 clones, which we did side by side. The second most abundant peptide in NGS wasn't even found by picking clones. So you would expect to find at least the second most abundant binding by picking 100 clones, but we just didn't. So this is what you would miss if you only pick clones. Counting sequence abundance is also not enough. As we saw from our experiment, the binder that ranked 3rd in the NGS did not even show binding, whereas number 17 did show quite nice binding. So how much time did it take in total? For MPD, we usually calculate 9 days for a full 96 well plate. After the enrichment process, which might vary between 1 and 4 rounds, you do the qPCR and NGS, which is done in a single day, as well as the primer synthesis and in vitro translation, which is calculated with 3 days. We'll have 2 days for ordering primers, and 1 more day for the biophysical characterization. So the advantage of standard phage display versus multiplexed phage display is, first of all, throughput. I was able to handle 6 library reactions at a time. With multiplexed phage display, I was able to handle a full plate. And the proof of throughput you will see in the second part of this presentation. Monitoring of enrichment is done in plating out titers, whereas here it's qPCR, and you also already end up with your sequencing reactions. Sequencing is done in Sanger sequencing, and here NGS. So rather than receiving 100 sequences per library, we ended up with 50,000 sequences per library. Specificity, here it is determined by spot ELISAs. Here you can already use the NGS data and don't need to do any additional experiment. The binding affinity is sometimes done using phage titration ELISAs. Here we can measure absolute affinities using the strep-David infusion technology. All right, so how about throughput? And this project or this experiment really demonstrates the throughput of this technology. So here we show you a technology to bin and also map the linear epitopes of 96 antibodies. We have 41 individual clones, antibody clones, 7 controls, and everything was done in duplicate. And this whole experiment took only 2 days. And we used 2 complementary technologies for that. The strep-David infusion technology as well as multiplexed phage display. As an example protein, we use signal peptidase B, which has been identified as a target to treat Staphylococcus aureus infections. And its purpose is to cleave signal sequences. So SPSB, in short, has been injected into a mouse, and using standard hybridoma technology, 41 individual clones have been harvested from the mouse. What we now need to know is where do those antibodies bind on the protein? And this technology is really only for linear peptides, not for continuous peptides, not for discontinuous ones. No structural epitopes. And everything was done in duplicates. So the big question really is, like, where do my antibodies bind to? Because this is extremely important for any further characterization. So what you do is you start off with the strep-David infusion technology. So this was the sequence of the protein. And what you do now is you take 20 amino acids and you synthesize another primer. And you take the next 20, another primer, and so on and so forth. And in this way, you're covering the complete protein. So you have primers for every region of the protein. Then you do again the strep-David infusion technology, and you end up with double-stranded DNA pieces, peptides fused to strep-Davidin, having all the in vitro translation features. Then again, you do in vitro translation, and you produce peptides. And now you've basically your complete protein in peptides. What you do now is you take 8 octet tips and you cover and you load your epitope-specific peptide. And you basically do that for every peptide, for every epitope peptide. And in one run, you're going through the complete plate. So this means every antibody actually sees every epitope of your protein. So you don't even need a target protein. All you need is the sequence, and you basically produce peptides. So what you then see is, as you go through the plate, if an epitope basically matches the antibody, or let me put it the other way around, if the antibody recognizes the epitope, you will see a binding event. And what you see here is the analysis. So we basically now bin the antibodies into different epitopes. Sometimes you see 2 binding events because we designed 2 layers in order to also cover the regions where one epitope peptide basically touches the other one. So we just did 2 layers of peptides. This is why you can see 2 binding events. So what we would now like to see in the phage display experiment is an epitope that basically is right where the two peptides meet. So what you do now is in order to confirm the results we have seen from the streptavidin technology, you now coat the antibody onto the well. So now you have a 96 well plate, and you coat an antibody in each well. Now, since the antibody is immobilized on the well, you actually add a phage display library, a random phage display library. And what we think that would happen is that the antibody will pull down phage that displays an epitope-like motif on its surface. So every antibody essentially grabs the phage that displays a motif that resembles the antibody. And this was actually confirmed. So if we look at the antibody clone 4E7, this is the epitope where it binds. The bioinformatical analysis for this was done in tripeptides. So we chopped off the protein SPSB into tripeptides, and we're matching the sequence to peptides where we see a good signal-to-noise ratio. If we now combine the two analysis methods in order to confirm the phage display results with the ForteBio octet, we found from 41 antibodies, 12 have unique epitopes. And this was extremely helpful for the subsequent high throughput screening campaign. Because rather than testing 41 antibodies, we're down to 12. And remember, this experiment was done in 2 days because we didn't have to amplify the phage, but we just put it on top, washed it, eluted it, and used it straight for qPCR next-gen sequencing. So this is the end of the second topic, and we would have another polling question for you. So having seen this presentation, would you be interested in being contacted for additional information on the octet system here? General acknowledgments are to Bob Lazarus at Genentech, who was my mentor and gave me the opportunity to conduct this project in his lab. The SPSB team at Genentech helping me out with the antibodies, which I obviously didn't raise myself. The oligo team at Genentech, I needed over 1,000 oligos for this project, as well as the Life Technologies team, which provided me with a sequencer and a lot of support in the beginning to get started on the next-gen sequencing. These are the references where I took information from in this presentation, and now I'm available for questions. Thank you.
Thank you, Dr. Hengstl, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Dr. Hengstl, our first question is, does this technology also work for larger frameworks?
Hi, Alexis. Can you hear me?
I can, yes.
Excellent, thank you. So this technology also works for larger frameworks. The only thing that has to be validated is the qPCR. So we tested this technology with 6 different frameworks, and for instance, for one of the frameworks, it didn't work at all. There was something with the structure, the DNA structure of the framework that wouldn't let it to be properly amplified in the qPCR. I guess we could have optimized the DNA sequence, but bottom line is it does work, but the framework itself needs to be validated.
Now, our next question is, did you use any automation?
We did not use any automation besides the devices we have basically shown in the presentation. So we didn't really use any liquid handlers. All we used was multi-channel pipetting, and we I was really focusing so that this technology can be conducted by a single person. I mean, you don't even need a next-gen sequencer. You can really send that out to other companies. What you need to do is you need to prepare the library, but no automation is needed besides the octet, which was shown, and the next-gen sequencer.
Now, Dr. Hengstl, do you include only randomized peptide sequences, or do you find flanking linker sequence can affect binding?
So for the library design, we actually did include a glycine and serine linker to make sure that the peptide is properly spaced to the code of the phage. And we did the same for in the octet system. So when we basically fuse our peptide to streptavidin, we also include a linker there.
Now our next question is, how do you amplify libraries from multiple targets?
That's a good question. Yeah, first we were thinking of actually amplifying them separately, but that would essentially destroy the whole point of the technology. And when you think about it, you start off with a completely random phage display library. And so what you can really do is you can pool the libraries from your complete plate and amplify them together, because there's nothing going to be in there that wasn't in there in the beginning. So when you then continue your phaging process, you just take this one master library and put it on top of all of the targets. Because if there's any non-specific peptide in there, you will see it in the control. So you can really amplify it all together. Just pool your phage library, amplify it overnight, maybe a little bit longer because you need more phage, you need to cover the full plate, but no need to keep them separated. Just pull them all together.
Now, is there a limit to the size of the library? Because you had only 50,000 peptides in your library.
So in terms of, there could be some, I'm not really sure what the size here means. So first of all, the insert, we were limited. So I was basically provided a sequencer by Thermo. I only had to buy the chips, but they provided me with the sequencer. And I'm not sure what their size limit is right now. Back then it was around 400 base pairs, but I know Illumina can do like double-sided sequencing. So this really was the limitation to this technology. If you can insert longer inserts, then there is no size limitation. The other thing that could be meant by this question is the number of sequences you need per library. So what we found is, depending on the enrichment, it's helpful to have either between 10 and 50,000, and that's just a rule of thumb. With today's technologies, you can easily have like 100,000 per library, and this is really just a math thing. So if you have, for instance, if you can sequence 5 million sequences, and you have 5 different libraries, you would get 1 million. If you have 50 libraries, you only get 100,000. But in general, as a rule of thumb, somewhere between 10 and 50,000 is, if you see enrichment, should be enough. Yeah.
Now our next question is, could you use this technology using other NSG modalities, not NGS chip sequencing?
Yes, absolutely. The only thing you need to adapt is the primer sequence.
So if there are no, I'm going to give it just a couple more minutes for any other questions that might pop in. Looks like we have another one right here.
Yeah, I think we can also answer the other questions.
So let's go with number 10. Did you analyze if some of the peptides have an advantage during amplification?
Very good question. So there's actually literature around that, and people have also developed technologies to basically, so we know that there are peptide sequences which are preferred in the amplification process. And there is, for instance, droplet amplification, and there are some really cool technologies out there. But what we found is we can completely ignore that, because if it's only amplified due to a preferred peptide sequence, they would essentially drop out in the analysis with the octet. And this is why it's extremely important to have a large pool of lead candidates, because you really can't tell if this is a preferred peptide sequence. So you just take a bunch of lead candidates and you test them on the octet, and the octet will give you a clear answer if this is really a good binder or this was only due to a preferred peptide motif.
Now, Dr. Hengstl, can you comment on the sensitivity of the different Forte Bio instruments and the size of the peptide associated on a biosensor?
All right, let's think about that question here for a second. So in terms of sensitivity, there are different octet instruments out there, which obviously do offer different sensitivities. We have used the octet red system, which was sufficient because we had the peptide on the tip associated with streptavidin, so we always had the target in solution. But if I'm more than happy to basically forward this question to my colleagues, and I know that there have been peptide experiments conducted with the octet where you could turn it around. But for our technology, it was more beneficial to have the peptide on the tip. And in this case, you usually have a larger target molecule for the dissociation and association measurement. I hope that answered the question, but I will in any case forward that to my colleagues so they can give you a more detailed explanation for that. And there's one last question left here. Can you get qPCR results on the same day? And the answer is yes, you can get that. Just a little add here, which I didn't show in the presentation. You might want to do a quick buffer exchange, because typically, depending on what you use for elution, this might inhibit the PCR reaction. So either you do a quick dialysis for a couple of hours, or a different method to basically exchange the buffer. All right.
So if we have no more questions, we'd like to thank you again, Dr. Hengstl. And do you have any final comments for our audience?
No, but if anyone's interested in the technology, so the technology-specific question about phage display, feel free to forward them to my email address. And yeah, that's it.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today or those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would like to thank LabRoots and our sponsor, Forte Bio, for underwriting today's educational webcast. This webcast can be viewed on demand through March of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Bye.